Good evening, everybody. Thank you so much for joining us. Welcome to the first webinar for 2025 of the South African Association of Botanists. We use this opportunity to share some of the expertise in the SAAB community. We would like to remind you that during the webinar, please keep your microphone and your cameras off so as not to cause disturbance. But you are welcome to pose questions to the speaker. So you can do this throughout the webinar by putting your questions in the chat. You'll see there's a little chat function there. You can type your question in there, and we will attend to them at the end. And then you also have the opportunity to ask your own questions at the end. So we would really enjoy if the audience can engage a bit with the speaker at the end would be great. And just a little note for SAC, those who need SACNAS points for attending the webinar, please make a note in the chat, and our secretary will attend to that to make sure that gets done. All right. So thank you very much, everybody, for making the time to join the webinar today. I will hand over to Sandy Lynn-Steenhuisen now, who is part of the SAAB Council, and she will introduce our speaker for today. Thank you.
Thank you very much. Yeah, good evening, everyone. No, thanks for joining us. I see our numbers have doubled in the last minute, which is great. So welcome. Thanks for your support. And yeah, I have the great pleasure of introducing a friend and colleague. So Prof. Grant Martin is a Deputy Director of the Center for Biological Control at Rhodes University, and a research affiliate with the Afro-Montane Research Unit and Department of Plant Sciences of the Kwakwa campus of the University of Free State. And yeah, we've been working together for the last few years on temperate weeds up in the Eastern Free State. But I know that Grant started by looking at invasive species in the pet trade as well in terms of aquatic plants. And yeah, so a great interest in invasive species and the control of those using insects from around the world. So yeah, has traveled a lot collecting in order to test their effectiveness for biocontrol in South Africa and other countries, with a lot of international collaboration. So without further ado, I'm going to pass over to Grant, and we look forward to his talk.
Cool. Thank you, Sandy. Thank you for the introduction. I am at home this evening, so if there's any screaming or squealing in the background, it's my children, and I apologize. On the whole, they are well behaved, but I hope they will be well behaved. They're often not. But I do want to take the opportunity to thank SAAB for the opportunity. I am a regular publisher in the SAJB, so it's nice to get another opportunity to contribute to the society. Yeah, today Nina and Elizabeth asked me to speak about the status of biocontrol in South Africa, which I will try to do to the best of my ability and where appropriate for my skills and for the areas where I work, which is specifically in the mountains and in the grasslands. So without further ado, I'd just like to give you a quick background on how biocontrol effectively works in South Africa. So there are two main branches. The first is the Agricultural Research Council, Plant Protection Research Institute, and their Weeds Division, probably more close. And this is a government branch that works on biological control. And I've listed the weeds there they particularly focus on: Mexican weeds, vines and climbers. They also have researchers working on pathogens down in Stellenbosch, as well as water weeds. And the second main branch of biocontrol in South Africa is run by the Center for Biological Control, based at Rhodes University, which was established in 2017 from what used to be the Rhodes University Biocontrol Research Group. And this leads back to about the 1970s when biocontrol first started at Rhodes University, led then by Professor Clan, who many of you would have come across in your time. And he started looking at citrus pests and cactus biocontrol and started a number of collaborations with the ARC. Rhodes University and the CBC specifically is now considered probably at the foremost of biocontrol research, both in South Africa and internationally. So that's our building down in Grahamstown at Rhodes University. And it's more than that now. It's a consortium of universities, and we fund both the salaries, bursaries, and running costs of various projects. So you can see on the map here just where some of our associated sort of universities and industry to the CBC are based to conduct the research we do. So we currently have 39 staff members, 45 postgraduate students, and 14 postgraduate fellows. And these are all funded on external income. So none of the CBC is funded by the university. It stands alone by itself. So the real aim of the CBC, and it's sort of what we aim to achieve as a center, is to try and reduce the amount of harmful pesticides going into the environment. And effectively within the CBC, we have two main branches. We have the insect pest side, which is the agricultural side. And within this, we work on bioprospecting for entomopathogens. With those entomopathogens, we conduct efficacy trials, we conduct field trials, and develop products all the way to where they can be used by industry. So effectively, it's the entire pipeline from discovering the organism till getting it out for mass production. And on the weed side, it's very similar. We develop new biocontrol agents for invasive plants. We release those biocontrol agents. We conduct post-release evaluation on them. We then follow that up by doing cost-benefit studies on those to try and show the benefits that they provide. We also do a lot of work, international work for countries who use our skills that we can outsource. And within the CBC, we have three main programs we really focus on. There are a few outside of these, but we particularly focus most of our resources into research into water weeds, cactaceae, and trees. And trees is very much the side where I've been doing most of my research. So moving to South Africa. South Africa has a few records we shouldn't be too proud of. To start with, we have at least 773, probably nearing 800 plants that are introduced into South Africa and have naturalized, meaning they now are self-perpetuating and growing naturally. Of those, 400 are 379, but leading up to 400 are declared weeds. That effectively means there's legislation stating that these are particularly problematic and we need to be doing something about them, and management should be implemented against these species. It doesn't mean it always is, but they are legislated to be controlled. Sort of another record we shouldn't be too proud of, and this is more appropriate to the side of research I've been working on, is South Africa has several times more invasive alien tree species per kilometer than anywhere else in the world. So I just want to show you quick what we're talking about in South Africa. Everyone knows, and specifically the current audience of today, the fynbos is really a biome we should be most proud of. A huge amount of endemism and a really high number of species. And we should be seeing these kind of vistas and experiences within the Cape Floristic region. A lot of small, colorful plants, a little bit prickly to walk through. But in actual fact, if you start doing descriptions of the vegetation, it looks more and more like this. This should be a fynbos vegetation. And instead, what we're seeing along the river course here, the sort of darker tree shrubs are, and then moving up onto the banks, there is a number of eucalypts invading. And these are both tree species introduced from Australia. We often get these blankets of pine species going up the hill, down the next hill, and up the next hill, the valley afterwards. And in this picture, you can see the mammoth task ahead of these gentlemen trying to clear this never-ending carpet of invasive pine species. Again, if we're talking about the grassland biome where I'm based, we should be seeing these vistas where everywhere you look, it should be grassland. I always tell my students when we're lecturing on this side of the world, if you look out the window and you see a tree, it shouldn't be there. But instead of these vistas, we're often getting sites like this. Again, Australian eucalypts lining a river, wattle species dominating landscapes, covering a huge amount of the land within the grassland biome. If we move out of the flatter grasslands and into the mountains, this is a nice picture of Golden Gate National Park that still maintains a lot of the grassland ecosystems. And as you can see, it's primarily grass species. But more often than not, we're coming face to face with these sorts of scenarios where it's been invaded by a number of northern temperate weeds. You can notice all the red berries in the front there. Anything with a red berry in the grassland shouldn't be there. It's usually coming from Europe and other areas of the colder areas of the globe. And the larger trees in the background there are poplar species also invading into the grassland. This is a population of invasive trees, Robinia pseudoacacia, that's invading a number of areas of the grassland. This is outside Clarence, close to where I live. And you can pick up a few other invasives, the poplars in the back and the willows in the river, but really a massive monoculture of invasive tree completely displacing the native grasslands. And a number of our rivers, to anyone who's accustomed to the grasslands, would be seeing these rivers completely covered with willow species, often quite, especially in the Free State, we like our willow species, but they can become problematic and dominating our rivers. These aliens really have a number of environmental and economic impacts. We can really get into a variety of impacts. Just about every species has its own unique impacts. Most of them use a lot of water. And they effectively cost the country a huge amount of money to try and manage. And they are starting to take up more and more land, potentially more than 20 million hectares in South Africa, producing water runoff from wherever they are really invasive. And it's estimated that this is a percentage of our standard GDP, probably in around of about 3 billion per year is being cost to South Africa by invasive species. I don't want to touch on too many impacts, but recently I've been working on some pines in the Western Cape. So I'll touch briefly on ones to highlight what they are, but they really impacts that everyone would be aware of. So I showed you some photos earlier about the never-ending carpets of pines, and you can see them again here. And in the middle is this global city of Cape Town. And years ago, we were faced with this potential catastrophic event of day zero. Effectively, city of Cape Town was going to run out of water. Thank goodness there was sufficient rains and refill. But when investigating what were the lead causes to this, invasive species, particularly pines and acacias, were really shown to be one of the drivers leading to this lack of available water for use in the city of Cape Town. Post that, a huge amount of resources have been gathered together. We're talking millions and millions of rands to try and clear the invasive species out of the catchments above the dams in the Western Cape. The same species, fires, and again another resulting in fires. And again, another example everyone would be aware of, the fires that impacted Knysna and the garden routes in 2018. Really having devastating effects, both damaging houses and burning people's homes down, but even worse, costing a number of lives. And this was again directly related to escaped wilding pines that are invading the fynbos biome. This is not restricted to the fynbos. The picture in the top right is my house at home. And in the grasslands, we're also struggling with invasive species, not always alien, both native and alien species really becoming dominant in the landscape, causing these much bigger fires that are much more difficult to manage. Luckily, though, for South Africa, we have had a very progressive government, and we are quite unique around the world. Not many governments, if any, put as much emphasis or have put as much emphasis and resources towards the management of these invasive species. Yes, we have a big problem that really needs to be addressed, but we are lucky that government has noticed these problems and really has put resources towards it. This is very much thanks to the late Carter Asmal, who you can see in the image there, who started the Working for Water program in 1995 with a measly, what we would think now, 15 million rand towards the program to start clearing invasives and to start researching how to manage invasives. And over the 25 years post that, to 2017, where we were starting to invest about 2 billion rand a year into the management of invasive species. If we average that out, it's about 300 million a year has been put towards by our government to the management of invasive species in South Africa. And that is really something. The program also has a dual mandate of trying to provide employment opportunities. Everyone knows in South Africa we're also struggling with large unemployment, so it's a really good initiative where we can reduce invasive species, but also offer employment, and specifically employment to women, people with disabilities, and the youth. So if we look at that program, how it's looked, and these graphs come from some work done from Brian van Bulchen and available in his Biological Invasions book, it just really summarizes the three main contributions of Working for Water. Firstly, the amount of money that's gone in, the number of jobs it's created, as you can see down on the bottom right, the bottom left, but on the right of the graph, employing over 20,000 jobs a year and clearing thousands of hectares of invasive species in South Africa. And it's estimated that this work has really been beneficial towards South Africa. About estimates getting up to $320 million US are saved every year by this program. And if the program hadn't existed, it would be costing us, the invasive species cost to South Africa would be significantly more than what it is already. So within the program, there are three main ways the people employed within the program are managing invasive species. That's mechanical, which is the basic chopping down, cutting out, pulling out of invasive species. Chemical, spraying with herbicides. Biological, very much my side. And then integrated control, which is any combination of the above methods. The first two are quite self-explanatory, but I'll just for a second, if you allow, I'd just like to explain biocontrol to people who are not aware, because a lot of the talk following this will be based on the fundamentals of biological control. So how biocontrol works is invasive species leaves its country of origin. In this case, in my little image, I see it's North America and gets introduced into South Africa. Sandy mentioned earlier I worked in the pet trade, so I'll use an example from that. I worked on a weed called Sagittaria platyphylla or Delta arrowhead. It's an aquatic weed, and it was introduced into South Africa coming in probably a small packet or as a seed, and then grown in South Africa. When it left North America, none of its associated organisms were probably with it. No insects that feed on it. And so when it got to South Africa, it was in an enemy-free space. It also then was released out of a fish tank into our water systems in South Africa that are now very similar to the water systems where it came from, which are slow-moving water systems in the USA. South Africa predominantly has rivers. However, we've dammed those rivers. That means we have slow-moving water, but no plants that are specifically adapted to growing in slow-moving water. And so it's not only escaped all its organisms, it's also found a habitat that's rich in nutrients and without many other plants there. So what I do as a biocontrol researcher, I go across to North America and I look at the organisms that are controlling it within its natural range. So there's not a weed there. It's only in South Africa that it looks like it's a weed. Anyone from the doozy might recognize it. It's starting to replace water hyacinth as one of the really dominant species in the doozy. I then bring back that insect. I've studied it in the native range. I consider that it looks like it could do damage to the plant. I then bring it back into South Africa where it goes into a quarantine system. This is insect jail, effectively. And then for this organism that I brought it back in 2010. I worked on it for 12 years to prove that it was specific. So I conducted host specificity testing on it to prove that it was specific to Sagittaria, that it could only survive on Sagittaria platyphylla, and that it could not do any damage to any indigenous South African species or any economically important species. Once it was deemed to be safe, I submitted release applications to our government who double-checked all my science. They asked international reviewers to check all my science, and then a decision was made whether they believed the insect was safe. Once that was approved, I was allowed to release the insect. And then I'll give some examples of what can occur in these situations. This is a particularly nasty cactus invading the Northern Cape called boxing glove cactus. And it's a species we've worked on recently within the CBC. I mentioned we worked on cactus and waterweeds. And we released a little insect onto that. And for me, I like looking here just at the tree in the background. In the back right here, there's a tree. If you look at its stump, and then it's still there in the background, but you can see the cactus is not looking very healthy. And within a few months, the cactus is basically just about all dead and dying and leaving the environment. Also worth noting in this picture is it's no longer a monoculture, and you can see the recovery of grasses coming through that cactus. Very similarly, this is a waterweed. I said we also worked on waterweeds, Kariba weed, Salvinia molesta. It comes from South America, from the Amazon. Again, really adapted to slow-moving waters, and found in every province in South Africa, really completely covering a number of our water bodies. And we released that little insect in the top left corner called Certabagus salviniae. And often what we'll get within a few months is this massive reduction or collapse of the weed cover. And on the whole, it will remain clear with only a small percentage of weed remaining in some of the smaller areas of the dam. So within biocontrol, we like to sort of describe the success or the impact these insects have in the following 3 graphs. I've put ABC here. So if we, just to explain the graphs quickly, the curve going up through the middle of the graph is a weed invasion. So it gets introduced at bottom left and slowly fills available area it can survive in, for example, South Africa, until it plateaus as it's filled everywhere it can grow. So the bottom is time, and on the left is the extent of the weed. At some point where there's a little, where that dark line starts, we can introduce a biocontrol agent. Now, I've just showed you 2 examples where we've got huge reductions. That does not always happen. So we have a scenario where we slow down the weed and it never reaches its full potential. The second example is B. We introduce a biocontrol agent, we stop that upward curve, and we get this reduction, but there's still a significant amount of weed in the country, and that we can then try and control using some of the other control measures, both the mechanical or chemical. And then the last option is like the 2 examples I've shown you before. We release a biocontrol agent, we get this huge reduction, and down to a point where there's very little left of that invasive species in the country, and we don't require intervention for those species. This is not new science. We've been doing it in South Africa for 100 years, over 100 years. We've released 130 biocontrol agents. Impacts, meaning we've never released an agent that's done damage that was unpredictable to the species. In general, we get good reductions of weed populations. And I have an overall saving of millions of dollars. Why, without putting a value there, is because every year biocontrol agents out doing work for us, we are saving money, and the value that we get back from biocontrol agents increases every year. Okay, so within the Working for Water program, going back to the Working for Water program, we've invested 300 million a year. How can we measure if it was worth it? And there are 2 main ways we use in South Africa to sort of determine whether the investment has been worth it. The first is using the SAAB database. I think a lot of people nowadays are aware of iNaturalist. This is sort of what we used before iNaturalist, but for invasive species. Basically, it involved researchers, botanists, and particularly one botanist in particular, Leslie Henderson, who used to drive around South Africa and record every invasive species she saw, as well as taking in any data that anyone sent to her about locations and densities of invasive species. We would then have a large database of these invasive species that we could look at the data and analyze. So analyzing the data from the SAPIA database between 2006 and 2016, we were able to see that all the species within the database, unfortunately, were increasing and increasing quite rapidly. That was despite the 300 million rand we're putting into their management every year. However, there was a silver lining, and a number of the species where biological control, however, was being used, those species were reducing or not spreading as fast as the ones that weren't, that didn't have biocontrol as a management option. The second method of determining this was a national invasive alien plant survey led by Ian Kotzer down in Stellenbosch. And this was work that the Working for Water mandated to try and see if the investment was worth it. And effectively what this was using satellite imagery as well as small aircraft to fly the entire country and map from air the invasive populations. The biggest limitation of this method was a lot of the smaller invasive species which were being picked up in the SAPIA could not be picked up, and was limited to the larger, easier identified species. They did this work in 2008 originally, and then finished again now in about 2023. It took a few years to do the second survey, and this has recently, as in the last 2 weeks, come out the results of the study. And the results from this study look a bit like this, and it doesn't make for good reading. If we look at the acacia species on the furthest left of the graph, and the values on the left is the areas of condensed hectares. That's effectively, if you squash them all into a very 100% cover, you have 1 condensed hectare. And that's the change that we have between the two surveys. So over about a 10-year period. And acacia species in this example are spreading over 100,000 hectares over the last 10 years. And very similar for eucalypts, spreading quite rapidly. But if you go all the way to the right-hand side, you'll see some species are decreasing in density. Cactaceae species, as I showed you one example, but we have many examples, and so they with good biocontrol are reducing. Sorry, the little insects above show if the species in question have biocontrol. So all the species where we have decreasing populations in South Africa have biological control options for them. When looked more closely through the data, there is one point I'd like to raise, and that is there are a number of acacia species that are under control. They don't fall under the acacia main species, but the reductions in their densities are really limited to the Western Cape and the fynbos biome. And I'd like to just take you through examples of the successful biocontrol of acacia cyclops and acacia saligna are two you'll see, but I'll give you a few other examples. So the first example is acacia cyclops, and this you can see is a never-ending invasion of cyclops on the dunes of the Western Cape. And this is a species we've introduced biological control for. This is what the biocontrol looks like. One is a little weevil, a seed beetle that was introduced in 1995. This insect, both the adults and the larvae, do damage to the seed pods, which are presented there on the left, and you can see where the name Cyclops comes from. And then after that, we released a little sessidemyid midge, or a gallfly. And what that gallfly does is it lays its eggs within the flowers of the acacia saligna. Those flowers then don't develop into seed pods. They rather develop into that little gall you can see in the image. That prevents the plant making seed. The combination of those really reduce the seed load being produced by the species. The graph on the bottom right shows the seeds that are entering the environment per meter square under invasions of Acacia cyclops. I'm going to embarrass a colleague quickly and put a picture of her up. That's a colleague of mine, Kin Canavan, and she's setting up a simple seed trap. And what that does, it traps the seed rain out of the seeds, that's coming out of the canopy above the seed trap. And so for a lot of the acacia research, we have these either hanging below trees or installed on the ground, and we can catch the amount of seeds that enter the environment. So in the graph, you can see the seed rain quite high, over 1,000 seeds per square meter on the left-hand side. But once the gallfly and the weevil start getting established, moving towards the 2019s in South Africa, in 2019 and more recent times, there's absolutely no seeds entering the environment. And so with this reduction of seeds from the biocontrol agents, along with fire regimes, along with clearing, we are starting to get real reductions in acacia cyclops, as you can see from the national survey, when it's measurable over 10 years, we're getting a reduction of a tree species. So environments that used to look like that are starting to look like this. The next wattle species I have to talk about because it's the most abundant in South Africa and the largest distribution, and really a species everyone knows about is black wattle. And here you can see a nice invasion in the grassland leading into the hills in the background. So with black wattle, again, we've released a very similar set of organisms. They're not the same. Each wattle has its own unique biocontrol agents. And so we released this weevil, Melenterius maculatus, in 2008. And then we released a cessidemide midge again. And once again, you can see those galls, the real diagnostic galls that the midge has arrived. So anyone living in the Western Cape would now, if you have a black wattle on your property, you will never find seed pods. You will only find these galls as the cessidemide has spread throughout the Western Cape. And again, we have a very similar graph here. The 0 on the bottom on the x-axis is the date that that midge arrived at sites in the Western Cape. And then we have the seed rain in seeds per 1,000, seeds per meter squared. And you can see on average, it used to be in the 1,000 to 2,000 seeds per meter squared. But at 4 to 5 years after the establishment of this midge, there's no longer any seeds entering the environment, and suddenly management for these big invasive species becomes feasible. Longleaf wattle, this is an invasion here. All the yellow on the trees is an invasion down in the Western Cape. And we've released, again, not quite the same system, but a seed weevil. And then in this case, a little gall wasp, and this wasp calls these diagnostic galls all over Acacia longifolia. And here's the same, the before and after. You can see this yellow field in the top left, and then on the bottom image, you can see that field of Acacia longifolia is no longer within that system. However, if you look on the bottom of the road cutting through the image, you can still see some yellow in that system. So there's still Acacia longifolia in that, but it's massively reduced. And this really gives opportunity to get on top of the problem using in other invasives. I always include this example because it's somewhere I used to go jogging in Grahamstown. This is a valley just outside Grahamstown that was heavily invaded with Acacia longifolia. However, with the introduction of the biocontrol agents and 2 fire seasons, there's no more Acacia longifolia, and a lot of the native Albany thicket has returned into the system. And then this is the last example of acacias I want to use. Again, it's from the dominant for the Western Cape, where we're getting these reductions in tree population. This example is included to show they're not always insects. This is a rust fungus called Euromycladium. It affects only Acacia saligna. The graph down on the bottom right here is different. That's a percentage of the original cover of the invasive species in South Africa. The rust was introduced in 1993, and we started getting this huge decrease in tree populations to where it is now very occasional within the landscape in the Western Cape. I want to move now to the grassland. And again, looking at my little graph in the bottom right here, we didn't have examples where we had reductions of acacia species in the grasslands or densities. We only had an increase. And we currently have acacia species covering over 350,000 condensed hectares in the grasslands. And that's just acacia species. We've got a number of other species. Top left here is a Robinia pseudoacacia I already spoke about. And then in the bottom left is invasion of a native species, Leucocidia sericea, that's spreading out of the valleys and into the grasslands, as well as willows. And so we're getting this real woodification of our grasslands. So I'm putting in this graph just to show where we're sitting in South Africa. So again, we have the area invaded across the x-axis of this graph, then how much it costs to manage that area of invasion. And then the impact of the invasive species is sort of a linear relationship between them. But at some point, it becomes just too expensive to control these invasive species. And I very much believe for the grassland, unfortunately, if we're sitting here, if we're considering chemical and mechanical control, it's just too expensive for us nowadays to try and get on top of the problems we're dealing with. So sticking with wattles, the grassland is really very much dominated by Acacia dealbata. And it's something that seems to be spreading. It's definitely not one of the species we've got on top of, but we have worked on biocontrol options for it, and we've released a weevil like we did with the other programs in 1998, but it does not yet seem to be doing anything. There might be a problem with how much we've released it, and we need to get it further into the system. But we really needed to do a bit more. And luckily, we have been releasing additional agents. So just in 2016, we released Dasyneura pyrolifera. I spoke about these sessidimide midges that we released on some of the other species, especially black wattle. They've done really, really well. But those were released in the 1990s and early 2000s. This we've only released in 2016 in the Western Cape. And then I made the first releases in the grasslands in 2022. And we haven't even yet actually confirmed establishment in the grassland. I'm sure we will get it into the grassland, but it's going to take time for us to start seeing any of the benefits that they have witnessed in the Western Cape. In the same vein, we've released a second little wasp. I was also involved in this, and we released this just a couple of months ago. We made the first release in the grassland biome. It's a little wasp called Perilampella hectoranus, and yeah, holding high hopes that it's going to start having significant impacts on Acacia dealbata. The next species I want to talk about is Robinia pseudoacacia. It's a species I've worked on for a number of years now. It's a really troublesome invasive species all over the grasslands. And it's diagnostic by, it's got some really big thorns on it, and you'll probably recognize it by certain times of the year. It gets these big white pendulous flowers and really makes it stick out in the environment. Very prolific around Gauteng, the Eastern Free State, but also down into the Eastern Cape. What makes this tree particularly difficult to manage is any mechanical damage, chopping down or wood harvesting really stimulates a tree to sucker, allowing it to spread rapidly via suckers. And the more you harvest the suckers, the faster it spreads and the bigger its thorns become, make it particularly damaging. I'll speak about a little program we worked on in the Clarence Village Conservancy. I've worked with them for a number of years, and and the Clarence 20 years ago, investing into management of a few small patches of it within the town, using mechanical and chemical control. Over the past 20 years, the Clarence Village Conservancy put 10 million rand into trying to manage these small populations. And as you can see in this image, a number of stumps there, they were very clearly cut down, nice and low to the ground. And then the blue color on the stumps is to show herbicide was placed. But unfortunately, in the added little image, you can see the suckering that's occurring from that stump. And if areas where we would go and monitor the clearing, we would get these examples where you could see a number of years of clearing, but still suckers coming up from the ground within the area. And so with the investment of 10 million rand over about a 20-year period in the Clarence area, what started at a small couple of hectares has spread significantly, and we're estimating about a 400% increase in the cover of Robinia in the town. So I've been working on for about the last 5 years, looking for biocontrol options to implement against the tree, and we've really settled. We've looked at a number of different species, and at the moment we've settled on this little gall midge that's highly specific to Robinia. And what this midge does, it lays its eggs in the leaf, causing the leaf to roll, and then at high densities, all the leaves will then fall off the tree. This insect's probably not going to reduce the tree populations by itself, but hopefully will really slow the tree down and allow various other management initiatives which have already been implemented to maybe get a foothold and get on top of this tree species. I submitted the release application to this for this species yesterday, so hopefully within the next few months we'll get some good information back that I can get permission to release this agent and start assisting in the management of Robinia. The next example is also one we've been working on recently. It's willows. When I moved up and started working on tree species, everyone said, oh, you'll have to consider willow species. And so they were very much on my radar. And in South Africa, we have 3 main species. We have top left here is the weeping willow. It's an introduced species. And then the crack willow is bottom left, Salix fragilis, also introduced. And on the right is the native willow, Salix mucronata. And here's this image again that shows an invasion of both the invasive Salix in the Eastern Free State. A lot of work was done on the willows in the late 90s and early 2000s. No, a little bit earlier, sorry, the late 80s and early 90s. And willows, specifically Babylonica, was regarded as definitely the most widespread and problematic riverine invasive in the grassland biome, with a number of negative impacts attributed to them, including water use and bank erosion. And so therefore they were really being considered for various management initiatives, and therefore was very much on my radar, as I mentioned, to consider for control. However, looking at the invasions of willows in the grassland biomes, I started noticing a trend, and anyone who lives in the area or anyone visiting the grassland biome will probably notice a similar trend, that most of the trees I was seeing within the biome looked particularly poor of health, as well as very infrequently were we seeing any young trees that would signify a spreading population. Most invasive species are easy to pick up in the landscape, as you can see a few adult trees, but then surrounded by a number of smaller trees, which we weren't seeing for Salix babylonica throughout the biome. And so what I wanted to do, and we had a master's student look into this, was to look at what the populations look like now between when they were first regarded in the literature as being so problematic. So we were able to bring up a number of aerial photographs from the 80s. And if we looked at those aerial photographs, we found we could zoom in and quite easily see what was assumed to be, from discussion with landowners, Salix species within the river course. So quite simply, we then put a dot on those so we could count them. And then we had these aerial photos, which were about 7 kilometers by 7 kilometers of counted Salix species. And then we could redo it with satellite imagery of 2024. And zooming in, we could do a very similar little exercise. And already see this reduction of Salix species within this example. And then we could overlay them on each other. And so the green dots here are the Salix that were in this system in 1994 compared to the Salix in this system in 2023. We replicated that for a number of sites through all the Eastern Free State. And the results show that in 1984, there's about 65 trees per kilometer squared of the areas we're looking at, compared to 2024, when these were down to about 40 trees per square kilometer. We then surveyed around, looking, as you can see in those photos, what did they actually look like, the ones that are remaining in the environment. And effectively, we graded them from being a seedling to dead. And you can see it's very much a population as modern day Japan is leaning very heavily to much older trees and potentially on the way out. So I'm interested as a biocontroller, what could be causing this? There wasn't a formal biocontrol program. There isn't a formal biocontrol program against Salix. We were considering it. We looked at the insects associated with the willows in the grassland biome. And interestingly, we found there was a high number of insects on Salix babylonica. Of those insects, also of interest to me, a number were species not native to South Africa, but rather native to where willow trees derive from. And so a number of its associated insects had naturally made it across to South Africa, including a cool little tortoise beetle, an aphid, and a leafhopper. And so as a biocontroller, I probably would have considered these if I was looking at biocontrol options for the tree. We also found another species, which is a sawfly. This was quite abundant and had already been recorded on willow trees in South Africa. Why I keep this one separate and of interest was this species has been used as a biocontrol agent for Salix species in Australia and is currently being used as a biocontrol agent in Australia and a successful biocontrol agent in Australia. So in this example, I think some of these insects are acting, they weren't intentionally introduced as biocontrol agents, but are acting as biocontrol agents for potentially damaging invasive in South Africa. But I wasn't convinced that these were at sufficient numbers to be causing a population level decline without other management. But we also noticed every tree we looked at had these really noticeable galls forming on both the young branches, the main trunk and stem of the tree, but also where the roots were exposed, you could see these galls. So the trees are heavily, especially in the Eastern Free State, heavily impacted by crown gall. We haven't had it identified yet molecularly, but from morphological attributes, we think it's this Agrobacterium tumefaciens. However, we still need to confirm this. And so what I think is going to happen with Salix, we got Robinia and Pyracantha that's on this heavy upwards curve. Hopefully Robinia will start bending over in a few years, but Salix, I think we're in a position where over the next 5 to 10 years, we're going to get this huge decline in the population, especially in the Eastern Free State. So I have a few concluding points. So there is hope for the grasslands. I think the Western Cape implemented biocontrol a lot earlier than what we have, and they're starting to see the benefits 15 to 20 years down the line. And hopefully those benefits, we've made the releases, we'll continue to make the releases, and hopefully if I'm still around, I'll get to see them, but hopefully my children will definitely see those benefits that of the work we're doing now. Unfortunately, biocontrol isn't for all species. It takes a long time to develop biocontrol agents. And so we need to really prioritize which species we work on. And I've been doing a lot of that with a lot of support from the University of Free State, the QPair Lab, and the ARU. And we'll continue to do that work to really work on which species we must prioritize to make the greatest change possible. Biocontrol also always struggles with funding. And so finding funding to work on the grasslands and species that are problematic to the grasslands is always difficult. Water weeds are very topical and receive good support, but often we struggle within what we need. Historically, as I mentioned, we've had amazing government buy-in, and they were very much thanks to those programs, the Working Forward program. That funding, with the change of recent government, there was a change from the ANC to the DA, which has affected funding. But we're working as much as we can with the current government, but we need to diversify our funding and look at other funding opportunities, because I really believe biocontrol is at least the one solution that, when all funding dries up, will still keep fighting the battles for us. So I'd just like to thank everyone who supported me with all the research, and all this information I presented is really thanks to other scientists. I've just sort of hopefully doing them a bit of credit here. and to everyone who supports me in my research. Thank you very much.
Thank you very much. That was excellent and very, very interesting. So thank you very much for the effort and the time put into that. I see we have a question here from Becca who's asking, given how water strained our country is fast becoming, do you think our government would endorse a release application for a biocontrol agent on eucalyptus?
Yeah, it's a great question. A huge amount of the projects I run, I work on pines in the Western Cape, is dealing with the conflicts that go with the management of invasive species. And so you shouldn't say never. I agree, eucalypts are really bad users of water. And there's a lot of changes within the forestry industries. For example, in pines, there's a movement towards hybrids, and we can find insects then that are that specific that they won't go into the hybrids. There's a change often from one species to another, and so sometimes a eucalypt species might become redundant, and then it could be considered for biocontrol. But we need to also pick our battles on where we focus our resources, and spending our time fighting forestry is sometimes not worth the investment. But I also have to give a huge amount of credit to forestry, who have really been communicative and upfront and supportive of biocontrol where necessary. We've released on all the wattle species, which are big forestry silviculture species in South Africa, with the support of the wattle industry. We're currently working on pines with the support of FAAB and the forestry industry again. So we're not working on eucalypts yet, but let's never say never. And I'm sure we'll also get good support from them when the time comes.
Thank you. Yes, indeed. That is always a difficult dilemma. All right. I see there's a question here about from Prof. Dave Berger. Is there an environmental adaptation reason why successful agents in the Western Cape for acacias will not work in grasslands in the rest of the country?
Yeah, what you'll notice from, I try to put up the images of the distributions. A lot of the species are very limited to their Mediterranean climate. So it's actually a different species of wattle that's dominating the grassland. So deal bart is very occasional in the fynbos biome, but very abundant in the grasslands. Saying that, species like black wattle are found throughout. And I've personally done some climate matching models to predict whether the insects should be able to get into the grassland biome. And it was assumed they would be limited to the Western Cape, and my modeling predicted they would be limited to the Western Cape. However, I travel between Clarence and the Eastern Cape, and I effectively drive through the route we would want them to move, because there's a slow march of the agents out of the Western Cape into the Eastern Cape. And before you get down off the escarpment there near Cathcart is where it would leave sort of up into and effectively get up onto the grassland. And when we were making the releases of Pilifera in 2022, we found the agents for black wattle there. And then post that, I found them 30 Ks on and 30 Ks on and 30 Ks on. And so despite my own personal work that I was quite proud on, it seems like they are getting their way into the grassland and are surviving. I don't know how far or if it's just because there have been a few years that are climatically better than other years that's allowed them to spread. But there are some climate limitations potentially to it, but I'm holding thumbs that they're going to continue to spread further into the grassland biome.
Thank you. There's also a question of, do you have any comments on the biocontrol success on pom-pom to reduce the invasion in grasslands?
Yeah, it's not a program. I'm very aware of the program. That's a program that's run by the ARC PPRI. and as far as I've seen, it's still in development, as these things take many years, but they've made a huge amount of releases, and at a number of the sites where they've made releases, they're getting real reductions of populations, and the agents causes the plant to stop sending out these big purple flowers, and it goes down to ground cover. And I know they are still making releases, so I would direct that question to the PPRI to ask them for. I know you can send requests for agents if you're dealing with pom-pom, and they should be able to come out to you and assist with explaining the program. But I would contact Liam May van der Westhuizen, to throw a name under the bus. Is the lead researcher on the program at ARC, and she's very approachable, and I would contact her for further information.
Thank you very much. There's a comment here that you had quite a lot of focus on the Western Cape and a few specific species there, but what about invasions into grasslands of Mpumalanga, KZN, Limpopo, Gauteng?
Yeah, a number of the same species wrap around into Mpumalanga, and we have those same seed traps. Sorry, I should have said this long ago, but a huge amount of the acacia research, the agent development, the seeds, the seed traps are done by the University of Cape Town, led by Professor John Hoffman and two of his colleagues, Karin Kleinjan, who was in the image, and Fiona Impson. And they have traps throughout the country, and they monitor most of the populations, and they have data for reductions in seed rain up in those areas. There's a bit of a north-south bias in South Africa, and a huge amount of research is biased to the Western Cape, and so that's where a lot of the good data comes from. But you're 100% right that a lot of these invasives are problematic all the way through the grasslands and up north. But again, we're not seeing the reductions of populations up there yet. And I also believe there's a bit of a bias on release effort. The early releases were all made in the Western Cape. There have been releases up north, especially in Mpumalanga. There's also been really good clearing programs in Mpumalanga. But yeah, if you could pull up the papers by the UCT team, you'll find information from Mpumalanga in those areas.
Thank you very much. There's a question whether there is a study looking at agent performance along an aridity gradient. I'm not sure whether this is for the grassland specifically or where. Yes.
Yeah, I'm currently in the field working with people on DroughtNet program, and we set up droughts monitoring stations in KZN and here in the Free State and into Kimberley, and I was very excited to consider if we could do some form of biocontrol across what climate change and environments becoming more arid. I wanted to look potentially also at the biocontrol of solanum in areas where it's getting drier. I'm not currently aware of any programs within South Africa. I might be wrong, there might be, but I'm not aware of too many studies looking at it on a aridity gradient. They are currently running experiments, but it would be a very interesting question, and I don't know what the results would be. Obviously, there's a relationship there between plant stress and insect health. That would be very interesting, but I don't personally know of any studies currently looking at that.
Thank you very much. And then are there follow-up studies on the biocontrol agents after they are successful? So Yonela is saying she's curious whether what normally happens with those insects after they've reduced or eliminated the invasive species. Do they find another plant to feed, not necessarily invasive?
Yeah, in this regard, there are definitely some amazing long-term studies, particularly on Pantheon acanthin, and Kruger Park is one nice example, done by, and on Panthea rantiaca, done by Helmut Zimmerman. looking at the relationships between a biocontrol agent and the host plant. And with biocontrol, you're never going to wipe out the plant. We never get eradication. So how it works is the plant population builds up, you release an insect at some point, it finds the environment where there's a huge amount of its host plant and its numbers build up extensively. The plant then starts collapsing or reducing, and then the insect reaches really high numbers. It actually, during the host specificity testing, we confirm it can just about survive on that plant and none other. So its options are it dies. And effectively, it starts dying out, and this allows the plant to recover somewhat, and then the insect own population increases. And you get this predator-prey curve that reduces down to, in a perfect scenario, to the organisms living with this boom-bust sort of existence at about 5% within the ecosystem. So very, very infrequently will you get spillover onto other plant species. And then if you do get occasional spillover, because sometimes, for example, water weeds, we released an agent on Harderbeerspoort. The agent built up to such numbers that when the water hyacinth died, there were tens of billions of the insects on the dam. And they were being attracted to lights, and people were like sweeping them off their tennis courts. And in those occasions, you will find that insect on other plants just because they don't know where to go. But they won't feed on those plants, they won't reproduce on those plants, they will eventually just die out of the ecosystem. So yeah, very much within this study, it's survive on the target plant or die. Usually if it can survive on another plant, we would have picked that up in the host specificity testing, and you wouldn't get permission to release that organism.
Thank you very much. Appreciate those answers. Here's a question. Is there research on what happens to biocontrol agents after extensive fires in their range?
Within the CBC, we have effectively we'd call it a stochastic event. And so it's not just fires, it can be floods, it can be severe droughts, it can be big rain. It could be sometimes they do such a good job that they wipe out the host plant to a point where their numbers fall off. And so we have mass rearing facilities around the country. It's a really great program. We use people with disabilities to mass rear our biocontrol agents. And effectively it's insect farming. So we have the target weeds in really healthy conditions, a lot of nutrients, high temperatures, and we add, and so we grow them. And then, so if you get a big fire, or it happens more often in water systems where you get a big flood, all the insects get washed out to sea or out the system, the weed returns. We mass rear these insects, and then we send them, we can send them back to where we need them after a stochastic event. Within the environment, if we're not replacing them after a fire, for example, most insects have wings, and there's a certain level of self-dispersion. So some insects disperse better than others, but they will move slowly. So agents that move slowly, we often facilitate them, but there will be a slow recovery back into ecosystems.
Excellent. Thank you very much. I think it's a super interesting topic, and I'm glad that we had a lot of great questions. We are, however, coming to the end of the night. It's been a really, really interesting talk, and we thank you once again for the effort and the time to come and present your work. It's been really interesting.
Thank you very much, and thanks to everyone who took time out of their evening.
Thank you. Much appreciated. And to the audience as well, thank you very much for joining us. It's always great to be able to share the SAAB community knowledge. Our next webinar will be in the second semester. So the next event to look forward to would be the SAAB conference in July. So you can find information about that on, please go have a look. And yeah, I think I can close off. Need to make announcements that I don't know about. Everybody for joining, and please have a good evening.